Okay, we're back, we're live, we're here. It's exciting today. Research in Manoa, actually research in Kaka'ako, I'd say, be more appropriate. Dr. Peter Hoffman, a PhD, Associate Professor of Cellular and Molecular Biology and Handwriting. And Dr. Aaron Rose, who is a postgraduate student with a degree in the same.
Yep, molecular biology.
Molecular biology. And they are both at JABSOM, the John A. Burns School of Medicine in downtown Kaka'ako. And we're calling this let's see, Solutions for Crohn's and Colitis. Is that fair enough?
Yeah.
Okay, and they're here to tell us about their research together. Let me put it in perspective. Peter is the mentor, the associate professor, and Aaron is working under him and doing some research on Crohn's and colitis. So the first thing that we need to do is define Crohn's. What is Crohn's, and why do you say Crohn's and colitis in the same breath that way?
Yeah, maybe I'll start and then hand it off to Aaron. But so it's an idiopathic disease, Crohn's and colitis. They make up the inflammatory bowel diseases. And while they share some symptoms, the location of where they happen in your GI tract is slightly different, and how they might be treated. In fact, how they are diagnosed and prognosed, and then how the MD decide to treat them are very different based on the two. And so maybe you can give a little more description of the two.
Well, I think that's a good breakdown. So it falls under the umbrella of inflammatory bowel diseases. We were talking a little bit earlier about what people are going through. I think, you know, the early characterizations of these diseases were kind of simplistic. You know, people would imply that you needed to run to the bathroom or you had bad gas or something. But it's
irritable bowels.
Yeah, and it's actually, you know, much more significant than that. You know, it involves ulcers and bleeding into the intestines. You know, as we've worked on this project, I've had the opportunity to meet people who actually suffer from these diseases, and you realize, you know, it's a significant impediment to normal life if you need to run to the bathroom 10 times a day and you run the risk of having a flare-up where you're in significant pain and you're bleeding, you know, and it's scary. And
what's the incidence of it? Are there a lot of cases around?
Well, there's about 1.5 million cases in the United States currently, and it's on the rise. So it's been termed a disease of the affluent. It's on the rise in developed nations very significantly. So around 70,000 new cases diagnosed each year, I think.
Okay, and we know that it has a lot to do with inflammation.
Yeah,
so can you describe the process by which the disease you know, takes hold on a biochemical basis?
Yeah, I can maybe start off, but essentially there has to be a commensal bacteria component. So if our guts were not an area where living bacteria were taking up, then no one would have this disease. So it's thought that we have the commensal bacteria in our guts. They're there for a reason. They help us digest. They also break down nutrients.
They're our friends. They're our buddies.
They can be our friends, and they actually keep away some bad guys from colonizing our gut. At some point, it's not known, but in certain individuals, there may be a crossing of sort of the safe zone. Laura Hooper at UT Southwestern calls it the demilitarized zone, the DMZ, between the gut bacteria and our tissues, and it's a safe zone that every once in a while the gut will experience a crossover. There will be some bacteria there. Usually, it's not a problem. Mm-hmm. And you can get a little bit of inflammation, and everything goes back to homeostasis. But in some individuals, or under certain circumstances, the crossover can lead to prolonged inflammation. Then the immune system comes in. It basically has to destroy a little bit of the tissue to get rid of the gut bacteria. That's an ulceration. And then if this doesn't stop and it keeps happening in a basically a remitting, relapsing disease, this is what ends up as inflammatory bowel disease. And it can go on for a long period of time, and unless you can get the inflammation under control, the immune system coming in there, destroying some of the tissue, and then it basically goes away, and then it happens again. you can end up with a long-term disease, starting often in your teens and keeping on through your whole life.
Okay, and you mentioned before the show that it was an ulcerative process and it involved fistulas. Can you talk about that?
Sure, I can talk a little bit about some of the more severe symptoms. So you can imagine when you have an ulcer, after that ulceration occurs, that tissue needs to be repaired. And if the ulcer is too damaging, or if the repair processes are hampered in any way by continued inflammation, then you could have the growth of the new tissue move into other organs, move towards the skin. So in some cases, you actually end up with physical attachments between the bowels and other organs, and the bowels and the actual skin of the patients. I've seen
Or parts of intestine to itself.
Sure, absolutely, wrap around to itself. And that's why surgery is so often an endpoint for the most severe cases, because of fistulas and because of thinning of the tissue. With constant inflammation, long-term flare-ups, you can end up with really thinned bowels, and then you run the risk of sepsis with tears in the bowels.
Sounds terribly, terribly unpleasant.
Yeah. I mean, in speaking to the molecular side of things, and Peter mentioned the demilitarized zone, it's very interesting, right? You have three sort of major components to what's going on. You have the makeup of the microbiota, you have that protective barrier, the mucins, the mucus that protect the self-tissue from the gut microbiota, and then you have your immune system. So you asked before if genetics played a role, and you can imagine if you have some sort of genetic makeup that causes your immune system to be a little bit overactive, that might cause the problem, because you could have your immune system overreacting to normal bacteria. Or if you have mutations that cause the mucins to not form their normal structure, you could end up with improper protection. You don't have that normal protective barrier that keeps things separate. Or if you have an imbalance in your gut microbiota, then you end up with maybe a bad bacteria growing there, outgrowing its competition.
What's mucin?
Mucin. So there's a couple of different mucins there.
It was a breakfast cereal, by the way. Oh, that was muesli.
That was mucilix, right.
Different.
A little bit different. So yeah, they're proteins that are secreted in the gut that build up that slick protection.
Okay.
They
Protects
provide
the epithelial.
lubrication, and a physical barrier.
Okay, so, and one last thing about the progression here is that this can lead to cancer. What about it is the thing that leads to cancer, do you know?
Yeah, and this this is not as well known, but there are several studies in humans that have basically human patients who've had this for long term, and the number is roughly 30 years. If you're living with this disease for 30 years, which a lot of people do, it increases at least sixfold, maybe eightfold, your chances of getting colorectal cancer. So it's not, the numbers aren't solid, but there is very likely an increased risk for out of control tumors growing, colorectal cancer arising. Colorectal cancer probably starts to arise in almost all of us at some point, but if you have prolonged inflammation in the gut that might be sending out some survival factors, it can give some of these small tumors an advantage, a growth advantage, and they can grow out of control. And so there is an increased risk. Exactly how that's happening is not known, but I have heard from some of my friends in the MD world who are treating this is that some of the new immunotherapies are not only interceding and intervening in the inflammation side, but they are very likely bringing down the colorectal cancer associations as well. So if you can treat the inflammation, it's very likely you can bring down the rise of the tumors.
Inflammation, big problem. Inflammation can create Crohn's and colitis. It can also create cancer. And if we can stop inflammation predictably, that would be a huge benefit for health in general, wouldn't it?
Yeah, it's tricky though, okay? So I told you I got to meet some of the people who actually suffer from these diseases. And so when they take some of these anti-inflammatories that are becoming really popular now, we talked about Humira and Remicade, it comes with a warning. So they actually are told that they may need to change their lifestyle in order to avoid potential infection. And when I looked at the clinical data, I really didn't see a significant increase in infections. But I didn't realize that we're kind of putting our thumb on the scales, right? We're telling people, hey, look, there is a potential risk, and therefore you need to avoid it. You need to actively avoid, exposing yourself, getting cut, being in situations where you can easily disrupt that barrier, protecting your blood from infection.
So how do you change your lifestyle then if you want to deal with Humira and with the other one?
That's a great question. I mean, when I read on, I like to go onto messaging boards to see what sort of suggestions patients offer to each other so that I can learn about their experiences and we can try and cater the development of a treatment to what people really need. But when I read on there, I think a lot of patients have the same question that you're asking, and there isn't a real good answer other than to be aware of it.
Yeah, and report the first signs of infection, and you can go and maybe get treated a little earlier with antibiotics before it gets out of control. So and kind of related to your question is inflammation really is a double-edged sword. Okay, we have to have some inflammation in order to start the process of clearing out pathogens like bacteria, viruses, parasites, and yeast.
The inflammation activates the body.
It's the first step in an immune response.
Okay.
And without it, for instance, there are genetic diseases where you can't mount proper inflammatory responses. Chronic granulomatous disease, CGD, is one of them. You're missing one or more of the genes that's required for this sort of oxidative burst that starts off inflammation. And patients with those are very susceptible to yeast infections. Their lungs are very susceptible. So we got to be careful when we always talk about, treating inflammation and going in there and really knocking it down, because it's really a fine line. You need to be able to manage it and treat it properly. And that's what, some of the lead compounds that we're interested in, we're hoping are not the hammers over the head like some of those really strong medications that are out there now.
To go back to the beginning of this discussion, so it's been called irritable bowel syndrome and all that, and I mean that's the sort of the homeopathic, the home remedy kind of name for it. But isn't there a connection between stress? You called it the disease of the affluent. Stress, the need to achieve, therefore the stress of not being able to achieve in the way you want. Meeting those internal psychological demands that your family and upbringing make on you, not succeeding, and therefore you get irritated, and the irritation creates the inflammation, and before you know it, you got Crohn's. Is there anything to that?
Yeah, I can maybe talk a little bit about it. So there are some strides we made. In the past we might have too generally or vaguely characterized stress, and a prime example are peptic ulcers. So at one point when you and I were growing up, all peptic ulcers were due to too much stress, and we had too much acid in our guts, and it turned out that that's probably not true. That the stress that we imagined we were going through, a lot of these peptic ulcers were caused by an infectious disease, Helicobacter pylori, a bacteria. And so with the gut, there probably are some stresses that might lead to higher susceptibility, but without those commensal bacteria getting through, and they might not lead to the ulcerations we're talking about. But definitely things, stress, we need to talk at a molecular level. Some of that stress results in things like IL-6, interleukin 6. It's a bad guy. IL-6 has now been implicated in several...
Is that a bacteria?
It's a protein that's secreted by cells in our body, interleukin 6, and it's secreted for the reason of basically senescent cells. After you need to heal wounds, you need to start off inflammation, you need to heal wounds and cause a little bit of death so that you can have wound healing. IL-6 is evolved to do that for us. But when our bodies are making a lot of IL-6, interleukin 6, and it gets through your bloodstream or it's acting locally in the gut, you can actually end up with chronic inflammation. It's been linked to a lot of things. Obesity, actually the fat tissue, the white fat, the bad visceral fat that is linked to overweight and obesity, it generates a chronic IL-6. It basically, your body is flooded with this wound healing protein it's throwing everything out of whack.
Not good for you.
Yeah.
And I think that's probably one of the essential phrases of this discussion, out of whack.
Yes.
You've named
Yeah,
about
you're
half a
right.
dozen things, and if they're all out of whack, you're going to get Crohn's and colitis. And so the research that we're going to talk about in the next part of the show is going to be to try to isolate which ones are, when they get out of whack, are most problematic, and how we can put them back in whack by some miraculous drug or therapy of some kind. How exciting. But don't get too excited, because then you get irritable. We don't want to stress anybody out here. There's Dr. Peter Hoffman and Dr. Aaron Rose of JAPSOM, and they're working on cell and microbiology things, trying to find solutions for Crohn's disease and colitis here in research in Manoa. We'll be right back after this short break. Take notes.
Aloha, welcome to ThinkTech Hawaii. My name is Josh Green. I'm the host of a program called Healthcare in Hawaii. I'm a physician. I work in the emergency department on the Big Island. I also serve in the state senate, which, please don't hold that against me, doesn't detract from my television program. We speak about all the big healthcare issues in the state. We get together on Tuesdays from 2 o'clock till 3 o'clock in the afternoon, and we try to talk about the most important issues in healthcare. This is a terrific venue for people to learn about healthcare. There are many programs on this station. We broadcast it later, not just on the internet, but also on OC16. Thanks for joining us. Please be informed healthcare consumers.
Hi, my name is Seymour Kazimirski. I have a show called Seymour's World on ThinkTech Hawaii. Our show is about opening minds and facilitating conversations. To tell you the truth, I have no idea what we're going to be talking about. I have no idea who our guests are going to be, but I guarantee you we're going to have lots and lots of fun. Aloha from Seymour's World.
Okay, Dr. Peter Hoffman, Associate Professor of Cell and Molecular Biology at JAPSOM. He's the mentor for Dr. Aaron Rose, also at JAPSOM, a postgraduate student in molecular biology. Did I get that right? Okay. And these guys are also with a company called Biologic. And it sounds like Biologic is an opportunity to actually try to capitalize, try to commercialize some of the things you've been researching. But before we get to exactly what you're doing, I do want to ask follow-up. So are you doing research in your own sphere only, or are you collaborating with others elsewhere? Because we find in this show that everybody who is doing research is collaborating with somebody. So I'm wondering who your collaborators are and how far and wide are they?
Yeah, so Aaron and I have been working together since he came in as a graduate student some 6 years ago. And so together we've been collaborating with not only other individuals, researchers at JAPSOM, the Cancer Center. We built really strong connections. And Aaron actually strengthened a lot of that because he's been publishing some studies in mesothelioma, also in colorectal cancer. So we hope that this tie-in with inflammation and cancers will get stronger as Aaron sort of develops out. And then both nationally and internationally, we have collaborators as well.
Okay, where'd you go to school, you guys, just so I can get that out of the way, too?
I actually did my undergrad in way northern California at State University, Humboldt State, and then I did my PhD work here.
Okay. Peter?
And back a long time ago, where I can barely remember, I got my bachelor's at University of Iowa. I came here and got my master's of science at Hawaii, and then I went back to the mainland at the University of Colorado and got my PhD.
Okay, so now, what exactly are you researching and how are you going about that project?
Yeah, maybe I'll turn it over to Aaron. Aaron really has taken the business part of this and developed it, so I'll let him maybe address it.
Well, I like to start the story back with Peter's sort of baby project. What he got his major funding for was selenoprotein K. And what he found was that selenoprotein K was actually being cleaved. It was being cleaved during macrophage activation. The protein itself was being cut. And we became interested in what was actually cutting that protein. We found that it was
What is a cut protein?
So when proteins are degraded, The first part of the process is being cleaved by a protease. And that's the definition of a protease, a protein, an enzyme that moves around and cleaves up proteins that either need to be changed functionally or need to be recycled for their building
It
blocks.
separates the pieces of the protein.
Sure, yeah.
Okay.
And the protein is a long chain that folds up into a three-dimensional structure so that it can perform whatever its function is.
Okay.
So you can imagine if you cut that three-dimensional structure, it no longer performs its function.
Does it perform other functions, or is it not?
Yes.
Can, yeah.
Ah.
And in fact, the
Changes its
protease,
function. Right?
the protease that Dr. Wang Shi from China found that was cutting cell K was calpain, and particularly it was calpain 2, one of the isoforms of this particular protease family. And that protease is really what led us off on this path. So we said, okay, that protease is turning on when a macrophage becomes activated. It's cleaving this other protein that we're interested in. Let's look at what else is going on with the function of that protease. And as we delved further in, we realized that in inflammation, what's happening is this protease is being turned on, it's cleaving a bunch of targets, and then it's being shut down by an endogenous inhibitor called calpistatin. And that's a protein that just kind of wraps across the enzymatic pocket that's doing all the cutting, like Pac-Man. And once you shut it down, the inflammatory process can kind of be stopped, or at least slowed.
So you got the proper amount of inflammation. And so you get a cleavage for a short period of time, and the body responds by bringing it back down. And that's what we were referring to as the proper amount of cleavage, except when, along with Aaron, we were looking in some human tissues of folks who actually had Crohn's disease or ulcerative colitis for long periods of time. They weren't shutting down that inflammation, and their levels of this endogenous inhibitors were quite high because they kept trying to turn it off, but they couldn't. And
But you found a way that you might be able to turn it off.
Well, that led us to the hypothesis that we could use a non-endogenous inhibitor, a small molecule protease inhibitor.
What's endogenous?
Endogenous is just what's normally expressed by the body.
Okay.
So we're
so a
using
synthetic inhibitor
so we're
to
using
help out the endogenous.
Okay.
Sorry.
No. I mean that.
I want you to talk in the language of science, but once in a while, ask
Yeah.
for a definition.
That's perfect. That's perfect. Yeah. So we used this small molecule inhibitor in a model of colitis, and we actually found that we were able to stop the production of inflammatory cytokines by macrophages and protect against the disease itself. And those results were pretty exciting. That's what led us to form the Biologic Hawaii company. And we hope to get federal funding in the form of an STTR grant in order to explore optimizing the drug itself and further testing the optimized product, and then looking to do toxicity and safety testing so we can take it through the preclinical realm and into the clinics.
It's an FDA thing.
It is, absolutely.
Oh, you're on the trail already then.
Well,
long
we're
trail.
very early on, and it's a long trail. Even just getting to the point where you can ask to put it into human beings is a very long and arduous path.
Yeah.
And sometimes it's not described as a trail, it's a bridge, because you have to go over the valley of death, they call it, which you have an idea and you start your company, and most of the companies fall into the valley of death and never make it back out.
But we're different.
Yes,
Yes.
we're hoping, we're
Well,
building
is
a
that
bridge.
the Valley of Death, a financial Valley of Death?
Exactly.
Okay, got
Exactly.
it. I mean, this is really exciting. This whole idea is only, what, a couple of years old?
Yeah. It's
And it happened here.
Yes.
Wow.
Yeah, there's a lot of great ideas generated at UH Manoa, at JABSOM, at the Cancer Center, and not all of them are turned into commercial successes, but we're going to give it a shot.
Will you still talk to us after you're multi-billionaires?
Of course.
Thank you. So how do you see this happening? Is it a microscope? Is it a, what do you call a more powerful microscope? How do you look and see and test on what's exactly happening down there?
Depends on which part of the process you're asking about. So if you're asking about the pathology or what happens as a result of the disease in the tissues, then we can see that under a microscope. You can take a tissue sample and you can look and see what effects the disease have had on the structure of the gut, the invading immune cells, the presence of the mucin barrier, et cetera. But when you're getting down to the molecular side of things, it's nowhere near that exciting. A lot of times you're looking at numerical readouts from instrumentation based upon a fluorometric or colorimetric assay, an assay where you have a change in color based upon a certain occurrence. So
You can't see the Pac-Man
no, that would be great.
doing cleavage, did you say? You can't see that.
Usually it's a marker of some sort of experimental protocol, like a band and a gel. You've seen some of those on CSI perhaps. Or, so a
Okay.
lot of times
We learn
you
everywhere.
have to design the experiment just right so that when you see the bands or the patterns, you can interpret the data cleanly. And it's biochemistry, a lot of it.
Okay, so, but you know now that this works. You know that you can, if done right, you can control the inflammation. And the inflammation is the critical piece of this. I mean, I was getting the idea from our discussion earlier that, you might be able to ameliorate the colitis process by changing the biota, by changing the mucin, by changing the autoimmune response, by changing, I forget I can't read my writing, but other elements in this process that leads to the ulceration. Oh, there it is, ulceration. Somehow, slowing down the ulcerative process. All of those are possible ways to deal with this, at least logically. But you're zooming in on one. That's the inflammation process. And this drug, what would we call it here? Special K?
Yeah, we haven't come up with a name yet. have we?
No name. And in fact, as
I
we
mean, the
go
breakfast
through,
cereal.
Maybe secret sauce for now.
sauce, okay.
that's a good one.
Yeah.
So we're zeroing in on using the secret sauce to stop is it slow down or stop the inflammation process?
I mean, it's an interesting point. So we were talking before about the drugs that are currently being used, right? And I mentioned that it's an intravenous injection. So you get this protein circulating, this monoclonal antibody. It's actually an antibody circulating in your blood that binds to mediators or signaling molecules that are allowing immune cells to talk to one another, to say, hey, look, I've got something we need to have an inflammatory response against here. Come on over and let's cause problems. And so that works, right? But if you're blocking all of the communication by that molecule, that is a general immunosuppressant, right? But what we visualize is that we have this compound that you take as a drug orally. It's diffusing in the intestines, and locally it actually goes inside of the cells themselves. It's not blocking the communication between, it's blocking the production of some of those communicating molecules, those cytokines that would allow the cell or group of cells that are beginning this inflammatory process, it'll stop them right there before they have to start communicating throughout the bloodstream.
Yeah, we envision delivering a more specific local inhibitor that doesn't affect your whole body, but can target just the tissue that's being inflamed.
We don't have that yet.
Those sort of delivery mechanisms are being worked on by big pharma already. So pretty much what we can do is we can put our small compound into their current research. And I think so we don't have to expend our own energy and finances to do that. It's already being done.
Okay, so so what is your, aside from getting approved, which is, that's a whole issue in itself, what do you want this to do exactly? I mean, how much fine control can you have, and how can you achieve that?
So we have a compound that works right now, quote unquote works, but we need something that's stable. So ours is relatively unstable, and that means that you'll end up having less total bioavailability. Bioavailability is the way we term the ability of the drug to get past the cells that are blocking its pathway in the intestines and the colon and the gut as a whole. And so we'll need something that's more stable so that it can stick around for a long period of time in the small intestines and the colon. And while we're making a more stable compound based upon the initial structure, we can address other things like making it a better inhibitor of calpain-2 if it works better.
Be more complete.
Exactly. We can also investigate if we want something that's irreversible or reversible. You can imagine something irreversible would have a more complete blockade of calpain-2, but it might also have more negative side effects. Whereas if it's reversible, it can work when it needs to and maybe function less when it doesn't need to. So there's a lot of things that we can alter chemically, but yeah, ultimately it's about stability and the ability to inhibit calpain-2. Oh, specificity of proteases.
So what Aaron's company and that I've helped co-found, but he's really taken concern is taking this lead compound, we call it, and optimizing it. And that process is called lead optimization. It's done all the time in the drug world, and this is what Aaron's basically going to monitor and guide.
So you drawing in technologies from elsewhere.
We are collaborators who can use computers to model our molecular interactions, organic chemists who can build new versions of our drug, and then other companies that can test these basically on species for safety before they ever go into the first human clinical trial.
It's just like, you know, Thomas Edison trying all these different kinds of filaments, and you take one possible, you know, chemistry and you change it a little bit to another just a little bit different.
Tweak it molecularly.
Keep
right.
on going and going until you hit one that gets the right result. Is that the way it is?
It is, although we're no way we're going to compare ourselves to Thomas Edison because we have plenty of examples of folks who are doing similar things, and so we're sort of following their examples, whereas Thomas Edison was so innovative and paving his own paths that I would never try and do that comparison.
Okay. Well, so what's the timeline on it? I mean, you have a grant, that's a finite amount of money out there somewhere. Where does it go from here? Where are you in the continuum?
You know, we're at the very beginning. So in a couple of weeks, we're going to submit that technology transfer grant I mentioned to you. And that one could take anywhere from a year to 2 years. That will be about the initial chemical optimization and the efficacy testing. Once we've established that we have some really promising compounds, then we'll go back and we'll say, hey, look, NIH, I think we did, or NIDDK, I think we did a good job here, and what we would like to do is take it even further. They can do a phase 2 grant that it's about $1 to $1.5 million. And that would be our toxicity studies, so the studies that we need to do before we can really move into human beings. So I would say the whole time scale could run anywhere from 2 to 4 years. That might be being optimistic. I'm sure if you ask someone else, they'd say 5 years minimum. But then after that, we also have clinical trials, right? So we'll have to take it to people who actually need it, who the current drugs aren't working for, and see if we can get it to work for them.
And I might add that if there's any venture capitalists out there, they could actually shorten that time period by a significant investment. And of course, as it goes, they will get some payoff in the future.
Oh, sure.
That's part of what Aaron is doing also. He's entered in business competitions looking for venture capitalist money, angel investors, but also getting advice from big pharma. Some of the competitions that he's done here locally and in California, he's getting great input from
Well, can I plug them? They're Oxbridge Biotech Roundtable. They're the One Start business competition. I went last month. It was crazy. It was 2 days where we just sat down. It was in San Francisco at UCSF. And we just sat down in front of venture capitalists, in front of representatives from drug companies, chemists, and they all just sort of rapid fire, you know, gave us all of this information. It was very intimidating, but really, really valuable. I highly recommend if you're thinking about following this path in Hawaii, join that competition.
You learn as much as you give.
Yeah.
Okay, so you said that you need venture capital in order to get across the valley of the shadow of death or something.
Mm-hmm.
But why is that? I mean, what's the relationship of money and answering these questions preliminary to going for trials?
The Food and Drug Administration.
Oh, okay.
So we have come
protecting
up with
human health
That's right.
before drugs are put into them.
So the money helps you do what?
The money helps you to, I would say, in the stage that we're at, optimization, getting the best possible drug that we can, the best possible treatment that we can chemically. And then beyond that, the real expense is the toxicity testing. So it's about really crossing your I's and dotting your T's, showing what happens when you have increasing amounts of compound, not making the assumption that just what gets into the body from a pill is all that's going to get in, maybe doing intravenous injections, even if you don't plan on delivering it that way, to look for any potential side effects. So it's all the testing and it's all the manpower and work that goes into doing the best we can to predict potential side effects before we ever let it touch human beings. And even then, when we go into human beings in something like a phase 0 or phase 1 trial, we only really slowly ramp it up with, constant surveys about, have you felt any potential side effects? Are there, things of that sort.
And you can get people to be in the trials. You pay them a little bit of money and
For the very early on ones, that's often the case. But then beyond that, it's about going to people who aren't being treated by the drugs that are available. So that's the whole point of developing a new drug, is that there are people out there who are suffering from these diseases.
they'd be happy
they
to
have
be
no
part
option.
of your trials. Yeah. Now, one of the things, we talked about all these other possible, what do you call it, factorials?
Multifactorial disease that it is.
That's what it is. That's what this inflammatory disease thing is in your bowels. But can we be sure that solving the inflammation problem will solve the diseases? Or will other factorials then still exist to give you the disease anyway, inflammation or not?
It's a great point. So I think it highlights so Aaron and I are both actually what we consider basic scientists. We don't study science in the clinic. Neither of us practice clinical medicine. We do interact with clinicians, but we're really basic scientists. So we sort of, instead of coming at finding an effect in the clinic and then trying to figure out what's going on at the molecular level, basic scientists work the opposite direction. We tend to observe what's going on at the molecular level, and then we see something that's happening, being regulated in the body in a certain way, and that's how we come up with the idea to take it to the clinic. So I can say with a fairly high degree of confidence that yes, that stopping inflammation in the manner that we're targeting will eliminate most of the inflammation. There shouldn't be a compensation, maybe is what you're thinking of, where you dampen something and something else arises, because I think we're at the sort of the front of the series of events, so that we can really kind of get a key node in the network that is basically causing inflammation. We hope that that's what we're doing based on our basic science. So that's what we're aiming for.
Do you have any sense that you're actually in a competition? That there are other people out there looking at other parts of the sequence, of the factorial sequence, that they may get to the bank sooner?
Always, yeah.
For example, I mentioned before, I'm really not familiar with the details, but David Watermole, Cardax,
Yep.
Pharmaceuticals, and Axanthin, which is an inflammatory reducing drug, which I think he's in clinical trials now, am I right?
He is.
Could it be that something like that could intervene and leapfrog over your work? Is it possible?
It's absolutely possible. And I wouldn't say this directly to the investors. So if you're an investor, you can turn it off.
You don't have to, listen to this part.
But I mean, it's kind of a win-win game, right?
Yeah.
If we get to the market and people need and want our drug, then it's because people need it. And it's great, we won. If we get to the market and there's nothing there anymore and no one needs it, win. Everybody's doing fine and we don't have a disease to treat anymore, right? But yes, that is a possibility.
Yeah, and I think that the reality is that most diseases have multiple drugs available for any given patient. Oftentimes, there will be resistance that arises from a certain drug, so the MD has to switch them over to another drug. And in fact, in Crohn's and Colitis, some of the antibodies right now that are being used, very effective, very expensive, but people are building up a resistance to them. They're actually dampening the availability of those antibodies, so they have to switch off of those drugs. And so the more drugs that are available for any given disease, And the pharmaceutical companies do have multiple drugs in pipelines at any given time. So I'm not too worried.
Even the same companies.
Same companies,
Address
the
the
same
same
disease.
thing, but it's an
Absolutely.
alternative, yeah.
So we're not too worried that another anti-inflammatory will be competing against these others, because the drug companies actually welcome or different approaches for these therapies.
You also hinted at multifactorial treatments, right? So there's also potentials for combinatorial therapies. So if there is a concern that, yes, you're dampening this portion of the disease, but maybe you also need to treat with a drug, something that alters the makeup of the microbiota, you could couple that with your anti-inflammatory and take both treatments.
Yeah, so it's a cocktail. I mean,
Could
it could be a cocktail at the end. Just to clarify on the microbiota, I mean, I think it's very interesting, and not that I'm doing heavy reading on this, but I've seen and heard that microbiota is really a study of a lot of research these days, and sometimes your microbiota goes out of whack, and you have to put it back in whack. And we talked before about the possibility of simply removing your microbiota, or I guess,
Fecal transplants, they're called.
fecal transplant, yeah. And thus being able to control it. So you could, I mean, everybody has its own, unique. I suppose they could do that for identification, like fingerprints or DNA one of these days. Take the DNA of the microbiota and you'll have a unique individual right there. But I could conceivably go in on a genetic level. I could do gene splicing or something. I could change the microbiota and thus the effect of the microbiota, and hit this whole problem from another angle, right?
Yeah, I think so. I think in the clinic right now, you have to have a pretty severe imbalance of your microbiota for them to consider a fecal transplant.
Expensive.
And it's expensive, and it's fairly invasive. It probably has to arise from a fairly radical disruption of something in the body, and I'm not sure that that treatment would apply to the 1.5 million individuals in the U.S. who are experiencing this disease. You're right. I mean, I think that more and more these days, I think they are taking into consideration not just the body's cells, but our guests in our body. And I'm going to steal a really interesting quote from Laura Hooper at UT Southwestern. And she actually, when she talks, she gives these fantastic talks, and she's so tired of saying this because it's become so cliche, but we're only 10% human. If you take your body and put you in a blender and get you down to the cells that make up your body, 80 to 90% of those are bacterial. So, yeah, they're important. We always have to consider the bacteria that we're living with when we're considering developing new drugs.
I used to think when you met somebody, when you look at the world, when you enjoy the traffic on Bishop Street, you always have to remember that they're all mammals, and not necessarily driven by rationality or rational considerations. But now I'm going to have a different view of it. I'm going to look at them and say they're all bacteria.
Who's doing the driving, right?
Who's driving? One last question is that everybody knows that inflammation leads ultimately to cancer. And if you cure inflammation, if you can control inflammation, then maybe, just maybe, you can cure cancer. So is this in your career path, either of you? Do you see this coming?
Yeah, we're going to start some of that work now. We have some preliminary evidence suggesting that our drug directly inhibits proliferation of colorectal cancer cells. Interestingly, it's not just the inflammation, but it's the signaling pathway that's activated inside of cells as a result of the inflammation.
Communications.
Yeah, so when cancers arise, I suspect, from inflammatory events, some of the pathways that they become, we call it addicted to, for signaling, for constant proliferation that they're doing, constant growing to form things like solid tumors, they're tapping these signaling pathways. And our compound blocks one of those signaling pathways, and it seems that by blocking that particular pathway, we might be able to slow down the growth or maybe stop it altogether. So,
and may block more than just one, too. Aaron.
That's true also.
Aaron recently, he's getting ready for a publication that we hope will be accepted any minute now, where this compound we've been studying actually has a two-hit process. It not only dampens the inflammation, but it has a mild but important effect on the tumor cells themselves. So if we can get this drug to market, it may be more effective for not just limiting the inflammation, but suppressing the tumor cells at the same time.
It wouldn't be the first time that kind of thing has happened. You build it to do one thing and it winds up doing
Absolutely.
another thing. Okay, well, I wish you luck on that publication, and I hope you put everything in Biologic. Okay?
Yes.
And I hope you talk to me on the street again.
Sounds good.
Thank you. It's Peter Hoffman and Aaron Rose from JAPSOM doing incredible research. So Hawaii. Fabulous. Thank you very much.
Thank you.
Thank you.
Aloha.